Good morning, everyone. My name is Hans Petter Graver, and I'm president of the Norwegian Academy of Science and Letters. And it's a pleasure for me to wish you welcome to this year's annual event on the Nansen Neuroscience Lectures. The Nansen Neuroscience Lectures honor the groundbreaking contributions of Fridtjof Nansen to neuroscience. They are part of the Academy's annual Nansen celebrations. We're also having the Nansen Memorial Lecture this evening, so that's the second part of it. And then in the end of November, 30th of November, we'll have a conference on the League of Nations and the United Nations. And this, of course, is not only Nansen's contributions, but it comes also closely to another side of Fridtjof Nansen, which is very well known. Linda Bergersen and Jon Storm-Mathisen have been organizing these neuroscience events for many years, and we're grateful for them for organizing also this year's event on keeping the brain in shape. So please, Linda, the floor is yours.
It's a great pleasure for me to introduce Ragnhildur Þóra Káradóttir. She had a breakthrough in neuroscience in 2005 when she had a Nature paper while she was a PhD student at the University College London, where her supervisor was David Atwell. And from there, she got a lot of attention, and she was given the opportunity to go to Cambridge and start her own laboratory already in 2008. And since then, she has several breakthroughs, and she's been awarded the European Research Council Consolidator, meaning she is something that Europe wants to put attention to and make her the best scientist ever in the white matter field. And out of this, she's now a director of the Center of Myelin Repair, and it's myelin repair we are going to hear when she's giving her talk. And her talk is called Activity-Dependent Myelination, Mechanism of Learning and Brain Repair. Please, Ragnhildur.
Thank you, Linda, for the invitation and the scrutinizing, embarrassing me so much. But luckily, I'm behind my computer, so I can hide a bit. For now, I will tell you about activity-dependent myelination, and I think it is an important mechanism that may explain lots about our brain, how it learns, how we learn, and how we can use this mechanism as a mechanism of repair. So I will review first what we know about the CNS white matter, and then what we know about development of oligodendrocytes and myelination. And then I will go into regulation of myelination and focusing on the evidence for activity-dependent mechanisms, and look into some new idea called the myelin plasticity as they underlie learning, and then whether these mechanisms are in fact the reason why the brain can regenerate or myelin. So it was very early on in studies, more than 100 years ago, beautiful studies by Flitwick, that they noticed that myelin occurs quite late in the development in the brain. It's one of the latest parts of the brain to be occurring around birth, and then rapidly fibers in the full brain get myelinated in the early a few months after birth. It's a dramatic increase in myelin. But then that it continues all the way late into adulthood. And what we now know is that, in fact, if you look at the human brain, that it is, well, is partly indicated by gray matter, which is the computational output of the brain, but the other half of our brain is white matter, where information is being transmitted. And it's been shown that by dividing the brain into these equal parts, it is giving us the maximum computational power. Yet in neuroscience, we tend to focus more on the gray matter and so much less on the white matter. And I wonder what, in order to understand the brain, we really need to fully understand the other half. And in that half are oligodendrocytes that extend their processes and wrap around the axons in the central nervous system, producing this thick, compact myelin sheath that, in fact, lowers the capacitance of the axons, and that allows the propagation of the action potentials to go faster. But it's not just speed that myelin seems to confer. What I want to hypothesize here is that we know that neurons, when they fire together, they wire together. And then if you see this red and blue neuron here, that if they fire together due to distance, they won't arrive to the target neuron, the yellow one, and they won't depolarize it sufficiently for it to fire action potentials. However, one way to overcome this is by myelinating these axons differentially and therefore adjusting the speed of the propagation of the action potential, and in that way ensure that they target the yellow neuron at the same time and the neuron fires action potentials. I think that that is the important part of myelin. We know that myelin is important because when it's lost in disease, such as multiple sclerosis and many other diseases, it can confer both physical and mental disability. But what is quite unique to the brain is the fact that myelin is very efficiently regenerated, but not always, and that's why it's important to understand how it's regulated. But the reason why it's regenerated is because there are cells in our brain called oligodendrocyte precursor cells. And during development, they differentiate into pre-myelinating and then myelinating oligodendrocytes. So to understand how the brain functions in the white matter and regeneration, it's important to understand how these cells become myelinating oligodendrocytes. So how is myelination regulated? And I started my lab with this question, and my two PhD students, Arina and Iben Lundgaard, took this on. And Iben is now a PI in Sweden. So we know that axons in the central nervous system during development, and neurons send their axons throughout the nervous system. They are active at the time early in development, and 90% of these projection neurons are glutamatergic. This glutamate is being released around the time an OPC, so the oligodendrocyte precursor cells, migrate throughout the brain. And to understand what's happening early on, we record it from these progenitor cells in the brain of developing animals. And what we find is that from unmyelinated axons in the white matter, these cells get synaptic-like inputs, just like inputs between neurons, and this is happening in the white matter of the developing white matter. And we find that they express NMDA or respond to NMDA application, AMPA and kainate, less so metabotropic glutamate receptors, indicating that oligodendrocyte precursor cells have kainate, AMPA, and NMDA receptors. But when we record in a fully developed tract, there are very few in the brain that are fully myelinated. Oops. We don't see these synaptic-like inputs in the oligodendrocyte precursor cells, indicating that this seems to be a communication that occurs between OPCs and the unmyelinated axons. When we look at the role of these receptors and signaling in vitro, we take here dorsal ganglion neurons, seen here in red, and OPCs are placed on top, and they might differentiate into myelinating oligodendrocytes seen in green. When we did these experiments, what we found is that the OPCs, when placed on top, will differentiate and myelinate, regardless of whether the neurons are active or whether we block all the glutamate receptors. This is in line with the fact that we know that OPCs can even myelinate fixed axons or even wrap around nanofibers. So there's a part of myelination that has absolutely nothing to do with activity. However, when ureculin and BDNF are added into the cultures, or their levels increase, then glutamate receptors densities increase on the OPCs. They become sensitive, in particular NMDA receptor. They become sensitive to neuronal activity, and now together with neuronal activity activating these receptors, they initiate the myelination program in these cells. But what is key that this switches the cells and they myelinate. So there is a part of myelination that is activity dependent. we know now that that activity is now also being shown in vivo. We can, if TTX is injected in the optic nerve, you see less myelination. If you lower firing rate in cortical neurons with lowering their, hyperpolarizing them, you see less myelination. And conversely, when you excite neurons with channelrhodopsin or with using DREADS by depolarizing the neurons, you get an increase in myelination. So it's clear that the evidence is for activity-dependent myelination. This work and many others come to indicate our hypothesis is that during development, unmyelinated axons have presynaptic terminals that release glutamate, and OPCs receive these synaptic-like input, and that in the presence of growth factors, makes them sensitive to the firing rate of the neuron, and then if that's correct, they differentiate and myelinate. But this is all how we vision developmental myelination. But during development, even when myelination kind of is completed, there are these progenitor cells still present. And these cells are actually quite numerous. They are equally distributed here. So white dot indicating and a progenitor cells is labeling in situ for PTGF alpha receptor. You see it equally distributed throughout the gray matter as well as in the white matter. And this is in the adult brain. To see that a little bit further, if we go and use a mouse model where the OPCs have been expressing DS-RED, and we're using a 2-photon microscope going through the cortex of a mouse, a live mouse, you can appreciate that these progenitor cells in the cortex of an adult mouse are quite numerous, and they are in fact kind of connecting to one another. So what are all these cells doing in the adult brain? We've thought about this a lot, and we've come up with some thinking, is that it is known that new oligodendrocytes are in fact produced in the adult brain. It has been argued in the field that that may be mainly due to maintenance of already existing myelin. But old literature shows us from humans, and particularly Case in 1907, that looking at myelination through the cortex of the human, you see that it's increasing. Although, have to be said that these are post-mortem studies of people, one snapshot, and it's not clear yet whether the 60-year-old person had just better food supply, nicer life, whereas the 30-year-old may have had a more of a traumatic life and hence less myelination. To rule out any alternative explanation, so we capitalized on the fact that we can use model organisms, such as the mouse, to determine the pattern of myelination, keeping them in the same cage, giving them the same food and environment. And in fact, we see that in the mouse, myelination increases with age in the cortex. So there is some ongoing myelination in the adult. But what is really being highlighting the importance of myelin is in fact DTI imaging or magnetic transfer imaging, MRI imaging of humans, where you can image throughout the course of the human life, and some of these studies have been quite instrumentally done in Oslo, have highlighted an interesting phenomenon. This, alongside with old literature from 100 years ago, indicate that in fact myelination increases dramatically from after birth in the first 5 years, and then it increases slowly afterwards during childhood, and then increases again later in life and during the adolescence, and then still when we're adult is increasing but at a slower rate, but later in life it ends up decreasing. So in fact there are few different distinct periods of rate of change of myelin, one being at the time when we are young in the childhood, then the second one being during adolescent, and the third being when we hit more senior age. Now, when you see this, you start thinking of the incredible overlay with certain disorders known for the nervous system. the rate of change of myelin is hitting closely to neurodevelopmental disorders. The second greatest rate of change hits with the onset of psychiatric disorders. And the third dramatic rate of change in myelin hits with the neurodegenerative disorders. Now, I'm not going to say here that myelin is the cause of these disorders at all, but what I want to try to campaign is that potentially myelin may be underlying a range of CNS disorders that we previously have not considered, and it needs to be perhaps studied closer. And this particularly rapid changes in myelination may be linked to the fact that we have these progenitor cells throughout the brain in the adult. Well, these changes, are they due to that the cells are changing alongside with the environment? Are these different milestones causing any differences in the way that myelination is occurring? And one question that we wanted to ask, and these are a few of my PhD students took this on, whether all of these progenitor cells are all the same, regardless of where they are. And what we did is that we have transgenic mice where we have OPCs expressing a YFP protein under the endogenous NT2 promoter. And we recorded from these cells the early at E13 all the way to 330 or 11 months old animals. And here I've highlighted the milestones of myelination that are known and some of their important milestones in development. What we find when we record from these early cells throughout age is that early in development, they don't have any voltskitic sodium channels, that they acquire them at the peak of myelination and then they plateau. So it peaks and plateaus. Similarly, when we look at kainate AMPA receptors, there are none in the first time when they appear. They come up early postnatally, but then increase in conductance with age, as seen here. These are the mean data, and this is the population data of the cells, the proportion of cells with the receptors. Then when we look at NMDA receptors, they don't appear, and they don't appear until postnatally at the time when the OPC start to myelinate, and then they completely disappear in the older animals at the time when myelination starts to decline. So if we look at the closer on the population of cells, we look at the early time point. There are the E13 cells. When they're born, they have no ion channels, but they quickly acquire voltskitic potassium and kainate, and I have here kainate indicating both kainate AMPA receptors, as I'm not using the concentration, does not distinguish between the two. And then they acquire voltskitic sodium channels. But I want to note that not all of the OPCs at any time point are 100% having voltskitic sodium channels. Similarly, the NMDA receptors don't appear and only peak postnatally around in the first month, and then they completely disappear by 9 months of age. And if there's two peaks of of NMDA receptors, indicates the onset and the offset really of myelin. So look at where these different ion channel expressing cells have different transcriptomes. We did a bulk analysis of different ages when certain cells are in majority. So if you look at early embryonic, when they don't have an ion channels versus at the peak of myelination, of course, transcriptions that are related to myelination go up. Red is upregulated and green is downregulated, indicating in fact early progenitors are more focused on migration, the ones that don't have so much sensitivity to neuronal activity. Whereas when they acquire the receptors needed to sense changes in neuronal activity, they are more in a myelination phenotype. Then later in life, when they lose the NMDA receptors, they in fact lose responsiveness to activity, or transgenes that respond to neuronal activity. They lose their differentiation potential and regulation of cell cycle, whereas they increase in their relations to the immune system. So when to look at whether we can alter the cells by taking them out of the brain of the old animals and put them into a more neonatal environment. So we took neonatal OPCs and adult OPCs, put them on neonatal axons in a nice, soft, identical culture conditions. And what we found is that although the neonatal OPCs can myelinate, the adult ones really can't within time when they don't have NMDA receptors. So what we are hypothesizing, in fact, that we have this heterogeneity of OPCs. We have OPCs that don't have any ion channels, then they acquire voltskater sodium channels alongside kainate and potassium channels. Then they acquire NMDA receptors, and they lose the NMDA receptors and increase the kainate receptor densities. And what we think this is indicating from our transcriptome data and a lot of other data is that, in fact, we have states rather than heterogeneity, and the non-ion channel expression is more like a naive state that is important and can respond to different things. We have a highly proliferating state, and the cells with NMDA receptors are primed for differentiation, and the kainate may be, in fact, quiescent. Now, I want to highlight that in order to be states, that means that we would then be able to move between the two types of cells. And in fact, we are trying now to play with a lot of factors, and we think we can alter the states of the cells. And just to highlight the changes, if we think about it as states, they start off as naive cells, and this is in early time. But then
I don't understand any of this.
They
you see the audience? No.
No, I don't need. Why didn't you see the audience?
proliferate.
What is special about the audience?
Prime is increasing. Should I continue? And they increase with life. So with different areas of the brain, the states of the cells differ, with some having still like the early naive cells are always present. But then with age, the quiescent cells start to take on more predominantly and become the prominent cell type. So how this links to the two modes of myelination, we're still not fully aware of. But what we know is that the activity-dependent myelination mode depends on NMDA receptors, and that indicates that the time period for activity-dependent myelination may be limited by the different times of in the aging or in the developing mouse. And we know that perhaps it might indicate that early in life it's less dependent on activity, whereas at the time of adolescence, when we have the peak of NMDA receptors, that might be more dependent on neuronal activity. And in fact, we know that social isolation impairs myelination during the adolescent time period in mice. And that is the myelination that happens early during the adolescent time point. And if you knock out the ERB3 receptors for neuroculin, and therefore blocking neural activity, you prevent that type of myelination without affecting this myelination. So that will indicate that, in fact, activity-dependent myelination might be quite important for the adolescent type of myelination that happens perhaps with responding to environment and learning. And DTI imaging has, in fact, indicated us to think that myelin may have something to do with learning. For example, for children and during childhood, professional pianists, the thickness of the corpus callosum that connects the two hemispheres is in a linear relationship to how many hours they play on the piano. Whereas taking more adult people or students or medical students and PhD students and training them how to juggle with 3 balls indicates that there's an increase in the white matter tract that connects the visual cortex with the motor cortex, indicating that white matter changes are occurring with learning. To address whether this is actually has anything to do with myelination, Bill Richardson showed that when you teach mice on a complex motor wheel, you see a change in oligodendrocytes myelination. But this is motor training. We wanted to test whether, in fact, we can push this more close to training, perhaps, that we learned in school, and wonder whether myelin changes might be evoked by cognitive training. And this is done by Julia Bonetto in my lab, and Claudia. And we collaborate Tim Basim and Professor Lisa Saxita in Canada because they had a really good model that we could really test cognitive learning without having a motor component much to it. And to do that, we use a mouse model to start with, where we have Cre expressed in the progenitor cells. And then we also have a tau promoter driving membrane-bound GFP. So when we give tamoxifen, nothing happens until the cells differentiate into myelinating oligodendrocytes because they have the tau promoter, and therefore we get green myelin when OPCs have differentiated after tamoxifen into myelinating oligodendrocytes. So we use a working memory task. A memory task, it depends on the hippocampal and prefrontal cortex. So I really want to look at the synchronization between areas. And it's a test that reminds me a lot about tests that I played as a kid, memory, when you have to memorize the location of a certain images. The mice have a touch screen where they touch, see a light, and then they get a choice between two lights, and they have to identify the new one, and they touch with the screen. So if we a little bit go over this, the mice are shown these 5 boxes, and then they are shown one light, and then there's a delay, and they have to remember the location of this light, and then they get a choice of 2 lights. Now, I'm pretty sure you all have identified this as being the new one, so you would have gotten a strawberry milkshake, very sweet strawberry milkshake, so sweet that it is now not longer available in UK because of the sugar tax. But then if you get it wrong, you get the mouse gets a light for a short period of time, which they don't like. We set up the way that we have mice that learn the task, and then we have them paired to a mouse that goes in the box, does everything, but gets the reward or the punishment according to the other mouse. And then we have a home cage control that gets as much of milkshake as anybody else. Here's a little bit of video taken from the screen that we are looking at. Here's the mouse. These are the touch screens. It activates the task by going in here, and then it touches, and it has to wait, and then it gets a choice. This is the easiest choice. It got it right, so it's getting its milkshake here. So what we have is the mice who are paired don't really learn the association of the task, where the mice that are getting rewards or punishment according to how they're doing learn the task. And this is the easiest version of the task, and then goes harder and then harder. And this is the highest level. In these mice, we see that the new oligodendrocytes are made. And if we quantify this, we see that in the dorsal hippocampus and in the medial prefrontal cortex, but not in the motor cortex, there's an increase in myelinating oligodendrocytes in the eyes that learn the task, but not in the ones that are yoked or in the home cage. Now, to really look into where this is happening, we know that there's no real direct connection between the hippocampus and the medial prefrontal cortex. There are connections that go through the nucleus reunions or nucleus accumbens or amygdala and entorhinal cortex. And they have different associations with learning or memory. So we know that the nuclear reunions is associative learning, which is something that's similar to what we are doing. The nuclear accumbens is with reward-mediated learning, which is, of course, what we're doing. Amygdala is more emotional learning, and the entorhinal cortex more the navigational learning and spatial learning tasks. So we looked into the areas where we see myelination increasing, in fact, in the nucleus accumbens and nucleus reunions. In the areas there are, in fact, related this type of learning, but not in the entorhinal cortex and the amygdala. So myelin is really being made exactly in the areas that are needed for the mice to solve the task. Now, what we are really looking into here is some form of myelin plasticity. We know that axons can be partially myelinated, that they're myelinated, but then there are large bits of axons that are unmyelinated. And there's a lot of axons, say, for example, in the corpus callosum, that are unmyelinated in some areas, particularly frontal, is about 70% unmyelinated axons. So myelin plasticity is really recruiting these axons to become myelinated, and this is what we will be detecting, as well as internodes, the changes in between myelin on partially myelinated axons. The third type of myelin plasticity are changes in the existing myelin. And of course, this is not what we're looking at here, is what we look into. And the question, of course, comes, what is the role of this myelin plasticity? What is the role, what is the importance of having these extra myelin internodes being added in? So to do this, we go back to our model, but now we have inserted an animal by knocking out here myelin regulatory factor. So now when we give tamoxifen, we prevent the formation of myelinating oligodendrocytes. And if we look at this, when we knock out here myelin regulatory factor, you see that there is a less formation of new oligodendrocytes, whereas if you have the myelin regulatory factor, you produce myelin fine. So when we've knocked out the myelin regulatory factor, we see that there is a reduction in the performance of these mice. They learn the task less. This is just the tunnel versus the yolked mouse, and this is one that makes myelin and the one that does not make myelin. And if you remember the task being gradually getting harder and harder, we looked at using how many animals reach to the hardest level S1. And we see that the knockout mice, they may be able to learn the easy level, the third separation, but as the separation becomes less, they can't learn, and myelin is needed, therefore, to proceed to the next levels. So now myelin is this plasticity and may underline the way that we respond and learn strategies to solve a different cognitive task. So there is enormous plasticity in the adult brain in the form of myelin. And then I question whether this myelin plasticity is in fact the other side of the coin of why the myelin may be important and have a high regenerative potential, as we see, and how we can understand this mechanism by then harnessing that in order to improve myelin repair. We know that in MS, it's a demyelinating disease, it's the most common disabling neurological disease among young adults. The 2.5 million people in the world affected by this. And I have here a picture of Jacqueline Dupre. She was a soloist. Her career was cut short, just 27 years of age, when she was already one of the most sought after soloists. And so just to show how it is a sad affecting people in the prime. I also want to share with you that, in fact, MS might be one of our Viking heritage. From Saint Þóra in Iceland, he actually studied a lot of women with transient paralysis and made enormous good detailed description of them. And they reflect relapse remitting multiple sclerosis. This is in the book. It was though Norwegians who brought this to my attention. And what was interesting is that 1199, it was described in our parliament, their symptoms, and therefore something of great importance. So this has been with a disease that's been with us quite a long, and especially in our probably a genetic heritage. It is, however, Jean-Martin Charcot that was the first one who really tied the pathology to the disease and the symptoms. And here we have a section of a person who had MS, and we see here these, it's stained with a myelin stain. So myelin is stained dark blue, whereas neurons are light blue. And when myelin is damaged by an attack of the immune system, you get demyelination, and you see this very asymmetric lesions. And there are two possibilities. One is that the oligodendrocyte precursor cells are in our brain, respond to this lesion and repair. But their repair, for some reason, is always less thick myelin, and therefore is easy to be seen on the sections. Whereas if you are in the worst scenario is that when repair does not take place, and then you start to have a chronic lesion that is lacking a myelin repair, that leaves the axons vulnerable for neurodegeneration. And it has been difficult to distinguish between why you can have these two situations in the same person even. And it was thought that it was depletion of these progenitor cells. However, if you stain these chronic lesions, you can see that their OPCs are present here, the axons. So it doesn't seem to be more like lack of OPCs, but more a lack of differentiation, or block of differentiation. Now, we know that when myelin is damaged, these adult OPCs come in and remyelinate. We have different types of these cells and different types of myelination. So what governs myelin regeneration? Is it the default myelination or activity-dependent myelination? So to test whether remyelination is regulated by neuronal activity, I have a few postdocs who took this challenge or are working on this heavily in the lab. What we do is we take a white matter tract that is fully myelinated, one of the few white matter tracts in the rodent that all of the axons are myelinated, and that's a cerebral peduncle. And then we inject lithium bromide that kills off the glial cells, leaves the axons demyelinated, but because the neuronal cell bodies are far away, these axons are intact, and that's very important. And then you get full myelin regeneration after about 3 to 4 weeks. What is also good is that we have a very clear sequence of events. We have OPC, lithium bromide is given, cells are killed, and then OPCs migrate into the lesion, they proliferate, and then they differentiate. And this is all really well, clearly distinguished. Just to let you see it a little bit schematically, we inject lithium bromide. It causes the cells to die very focally, causes demyelination and activation of microglia. They clear out the myelin debris, and then you have activation of the OPCs. And that at this time point, we can go and record from these cells. And when we see that when we record in the lesion, the cells can sense glutamate, just like in early development, and only OPCs in the lesion are in development. Well, we see that the receptors that appear is that early on, we only have AMPA-K in it, like in development, and not so much NMDA receptors. As seen here is the proportion of cells. But within the second week, at the time when they differentiate, the NMDA receptor starts to appear, just like as when NMDA receptors start to appear during development. So it's phenocopying development. And as in development, and quite surprising though, is that these demyelinated axons that were fully myelinated regain synaptic communication with the OPCs. And if we look whether this is only a mouse-related or rodent-related issue, we collaborated with the MS Tissue Bank in London and asked them to stain for presynaptic terminals in human MS tissue. We're using the vesicular transporter for glutamate as marker of presynaptic terminals. And we see here in the lesion, there is a quite dramatic increase in the presynaptic terminals compared to in the myelinating areas. And they are quite increased, and it shows in these lesions we have OPCs that send their processes towards where these vesicles are, indicating that in MS, in humans, OPCs still seem to be communicating to demyelinate axons via synaptic inputs. So what's the role? Why is this happening? To do this, we put osmotic mini pumps, and the fact that we can put them into the lesion, and that if we put a dye in, we know that the dye remains within the lesion, so it remains very focal. And that allows us to be able to block these synaptic communication and look in what effect it has on myelin regeneration. And just to remind us that in the lesion, we only have demyelinated axons, macrophages, and OPCs, and only OPCs have glutamate receptors. So what we're going to be looking into now is that we're going to have cross sections of white matter tracts, and this would be how it looks schematically, and this is how it looks in real life. When the tract becomes demyelinated, you lose the contrast of the axons, and you hardly can see them. But when they get remyelinated, there's a thinner myelin that goes around them, and you can now see the axons again, but they're thinner myelin compared to here. But what we are going to be comparing is demyelination versus remyelination, so images like these. So when we block omega-conotoxin by using omega-conotoxin to block vesicular release of neurotransmitter, we see there's a reduction in remyelination. So indicating blocking vesicular release reduces remyelination. If we block AMPA receptors that are on the postsynapse of the OPC, we see a reduction in remyelination. And now if we take it further and we block neuronal activity, and here we have electron microscopy diagram where the remyelinated axons are in green and unmyelinated axons are in yellow, and you can really appreciate that there are fewer axons now remyelinated, indicating that activity is important for selecting axons for remyelination. Now, we've done this now multiple ways. We've blocked omega-conotoxin, blocked vesicular release with omega-conotoxin. We blocked the postsynapse with MPQX and MK801, so with AMPA and NMDA receptors. We blocked neuronal activity. We've also used DREAD to alter the membrane potential in the neurons that pass through the lesion by using reducing their membrane potential, so we're lowering the firing rate. Another lab, and all of this leads to reduction in myelination. Another lab has used generodopsin in the corpus callosum during remyelination studies and seen that increasing the activity increases remyelination, strongly indicating that the axon OPC synapse are necessary for remyelination. So our view of the world is that when myelin is damaged, if the neurons can remain active and upregulate synapses, OPCs enter into the lesion and they will then differentiate and myelinate. If, however, axons do not make presynaptic synapses or upregulate them or are active, OPCs still enter into the lesion, but they do not differentiate, but only proliferate, as they are lacking the synaptic input that are needed for remyelination. So where we are now is that we have think that that there are two modes of myelination: the activity-independent mode and activity-dependent. Activity-dependent myelin mode depends on activation of OPC and NMDA receptors. We find that OPCs are born the same, but they become heterogeneous. Heterogeneity is more likely reflecting cell states, and that the OPCs with NMDA receptors are in the primed state for differentiation. We've seen that cognitive learning evokes new myelin formation, especially in the brain structures that are needed to solve the task, and that demyelinated axons generate de novo synapses with OPCs, and that activity-dependent myelination mode activity-dependent mode for myelination is needed for myelin regeneration. So I will thank you for listening, and here was the Knowledge My Lab. We are now, of course, in lockdown for most of the year, so lab meetings have been on Zoom, and I want to thank my past lab members who helped with the work that I presented, and collaborators, and of course our funding. And I'm happy to take questions.
Thank you very much indeed. This was really impressive. I think you have brought up some entirely new concepts, and I think I'll first ask if anybody wants to comment or raise questions. Those in the audience will have to come up here to speak to the microphone.
Thank you for a very, very nice talk, Ragnhildur. You know, when you train your brain with Sudoku or crosswords or whatever, would it be even better if you did both physical training and Sudoku not at the same time? Can you comment on this?
Wow. Okay. I'm imagining when you say it, it's good that you said not at the same time. I see someone on the running wheel with a Sudoku in front. We know that physical exercise increases OPC numbers and proliferation. So that, and the physical activity has been shown to have an effect in that way. And most progenitor work or plasticity with progenitors in the subventricular zone, etc., has shown that physical activity is very positive towards that. The learning would increase all OPC's differentiation and myelination, but only in the areas where you needed it. So in the Sudoku areas of the brain, I assume. Whereas the exercise would, well, you're really pushing me in my difficulty. I think exercise is more cardiovascular effect, and the cardiovascular effect may, in fact, well, this is something that's fully hypothetical, or thinking, may in fact give the environment for extending the prime state. So if you're exercising, then your brain is more in a prime, environment that promotes a prime state. So therefore, myelination will be able to carry on for a longer period of time. So you might reduce that decline that we're seeing in the general population in myelination with age. So,
yeah, because I like this when you show this diagram from 13 to 20 years, and I'm worried, if the only thing that young adult people do is to be in front of a computer. So if this is we need that guidance, to tell what to do, to have the best brain ever.
Yeah, no, I think the exercise will give the environment, but I haven't done the experiment. Linda, we just have to do it together.
We can do it together. Thank you.
Very nice. On this same note, you had on one of your earlier slides showing that some growth factors, including BDNF, would be necessary to switch on the sensitivity of the myelin-producing cells to glutamate. And we know that exercise is one of the things that increase BDNF. So maybe there's a link there.
Yeah.
Anybody waving their hands? Yeah. Evandro Feifang, please come up here.
Thank you for your very informative talk. And we're also pleased to have you as a No-Age international member. I think from your talk, I got a lot of knowledge regarding myelin, especially one, like age-dependent changes of the wave. So it shows when people grow to 60 and above, there's a great reduction of myelin. And as you argued, it may also relate to neurodegenerative disorders. So indeed, last year, Professor Mark Madison, he had a paper in Nature Neuroscience, shows that A-beta can induce the senescent phenotype of the oligodendrocyte progenitor cells. And if you use senolytic approach to eliminate those senescent phenotype, you can improve the memory in the mice. And I also brought this idea to your colleague, Professor Robin Franklin, last year when I was visiting him. So I wonder, do you have any comments regarding this? Whether for the elderly people, we can do any ways to improve the myelin as a way to alleviate the memory loss? Thank you.
Okay, well, we've been trying to think about ways to do this, and this is why I kind of fleetingly made an comment on we are trying to alter the state, so altering the quiescent state into the prime state, and by either adding in factors that are needed or by eliminating. And I think it's very important if we look into, if we start thinking about OPCs as different states, then we can start to think about how can we prevent the quiescent state from forming and then move it towards more prime state and therefore prevent the myelin decay that we are seeing. Because I think the myelin decay is just a block of myelin maintenance that is happening in the brain.
This idea that you had on one of your first slides, that actually the myelin is adjusted in order to adjust the speed of conduction of the nerve impulse, so that the impulses from widely distant areas reach the targets, their common target, at the same time. That was very interesting to me, and I think a new thought. And obviously, for this to function, it requires really a sophisticated way of regulating myelination, which you have unraveled for us to some extent, but I believe there is a lot more to unravel. Can you give some thoughts about that very briefly?
Well, this has been a difficult one. How does the oligo know when the action potential is supposed to arrive at the right time? We are playing with the fact that the neurons knows. The neuron, that there might be factors regulated by the axons that are related to firing frequency. And that may be the way that it may be regulated, similar to when you think about the BDNF, again, on synapses, you can have them cleaved, and the TRK-B is trafficated backwards into the cell body of the presynaptic neuron. And we might be seeing some similarities in that. Alternatively, and this is very recent, is that the microglia might be involved with altering the myelin. And that might be activity-dependent alterations.
Yeah.
But I mean, this is all speculation. No one really knows how this is going to be happening.
It's interesting to speculate, of course. And this simultaneous arrival of impulses on one cell, I think, is a central concept in learning. So then you have sort of a circle here where you want to, how can these neurons know, or how can the myelination process know how to, what comes first and what comes after?
You need something that is fast, which is difficult. So, I mean, we ask ourselves this a lot, and we don't have a clear answer. My first aim is first proof that this is happening, and second, figure out the mechanism. But you kind of have to think about, you know, there are multiple avenues. Astrocytes are also one of my favorite explained, but I don't know the speed of their raise of calcium, whether they could be the ones that might be regulating it, and therefore regulating the microglial cells, similar to you see with synaptic pruning.
Yeah, it happens over. It's a process that will happen over some time, also not everything at the second time scale. And now I'd like to thank you very much. And I hope we'll be able to see you here in the Academy in the future. And now, thank you so much. And now Linda will introduce the next speaker.
I'm now pleased to introduce the next director. It's the director of Center of Healthy Aging at the University of Copenhagen. And Lene Juel Rasmussen, she has been leading this center since 2009. And this is an interdisciplinary approach to better understand aging, and especially healthy aging, and to redefine aging. Lene Juel Rasmussen she is doing a lot of things. And in 2008, she then joined the World Health Organization, and she their guideline for diagnosis of breast and colon cancer. That's just one of those things she's doing. she's in her subject of research. She's a molecular biologist, and she's working with how the cells repair themselves. So today, Lene will give a talk about healthy brain aging, understand and intervene. And having said this, she became a member of this academy in 2019. So please, Lene, I welcome you a lot.
Thank you very much for the invitation, Linda and Jon, and thank you very much for the introduction. I'll try to share my screen. Okay, so today I'm going to talk about aging, but I will have a specific focus on brain aging. But this slide illustrates why we are interested in trying to find sustainable solutions to this demographic change that we are seeing. We are right now at the crossroad where we have more people age 65 than we have people below 5. So that will give tremendous challenges to the society and especially the healthcare systems as we see them today. So therefore there are global efforts trying to find strategies to ensure what's often called healthy or successful aging. The way we define this is how people can maintain relatively low probability of disease and disabilities, high cognitive and physical capacity, and also be actively engaged with the surroundings, with life essentially. And we believe that the only way to really come up with novel solutions for this, you have to engage in interdisciplinary projects and collaborations. However, today I will mainly talk about some of the biological work we have been doing here in Center for Healthy Aging over the years. So it's clear that aging, as I said, it's one of the greatest challenges in our society at the moment, but it's also very encouraging to realize that there are actually coming more and more strategies how we can intervene of these age-related diseases. So that's also something I will touch on. And some of these strategies, they involved how we can prevent cellular dysfunction and decline during aging, but also restore function of aged tissues and organs, and even rejuvenate cells or tissues with drugs. So when we age, we'll see an increased disposition to a number of diseases, age-related diseases, from cancer to Alzheimer's and Parkinson's. And what is common for these diseases from a molecular point of view is that they are all characterized by some form of genomic instability and also mitochondrial dysfunction. And both these two are what's called the hallmarks of aging. So there are a number of hallmarks of aging which are characteristics of cells, aging cells. So that happens. So these systems, they are declining during age, but there's also a communication between these hallmarks of aging. So what I'm going to focus on today is to focus on actually the interaction of some of these hallmarks, mitochondrial dysfunction and genomic instability. So we know that there is an interaction because in some of the premature aging disorders, the phenotypes are actually defective DNA repair and mitochondrial dysfunction. So in vivo, it also suggests that there are some kind of connection between these two cellular pathways. And if we look at the genomes in these progerias or premature aging disorders, we see an increased accumulation damage load. So that suggests that they're actually not able to repair the DNA, and it's much higher than compared to normal cells. So that would suggest that if we can find strategies to actually decrease this genomic damage in the cells, that could actually extend the lifetime or the health of cells and tissues. And I know that there are many strategies at the moment trying to find small molecules to modulate DNA repair activities in the cell. So DNA damage is usually repaired by a number of DNA repair pathways. These are substrates specific, but they are also overlapping. And several of them, most of them actually have been linked to neurodegeneration in some form, especially the base excision repair pathway. So these pathways, they require energy to maintain the function of repair. And cellular energy is primarily produced by the mitochondria. So mitochondria provide bioenergy to essentially all tissues in the human body. Therefore, mitochondrial function dysfunction will have some serious consequences for the human physiology. So essentially, mitochondrial dysfunction can affect any organ in the body. So in Alzheimer's, it is also reported multiple defects of mitochondrial dysfunction. So fusion and fission events, mitochondrial fragmentations, bioenergetics, transport, movement along the axons are very important, especially to provide energy in the brain. And also another important cellular function, mitophagy, seems to be compromised. So mitophagy is a sub-pathway of autophagy that are some kind of cleaning systems in the human body. So these cellular pathways make sure to remove dysfunctional or damaged organelles, so in order to keep the tissue healthy. And that happens that in the way that the mitochondrial that the damaged mitochondria are engulfed in called a mitophagosome, which is fused to the lysosome, and then it's degraded. So a number of years ago, we tried to ask the question, so do we see actually DNA repair defects and mitochondrial dysfunction in Alzheimer's patients? So we did a small pilot study on Danish Alzheimer's patients and age-matched controls and measured various parameters, including DNA repair capacity and mitochondrial function in the blood cells of these patients and controls. And what we found was that, yes, there was decreased DNA repair capacity, and we also found several mitochondrial parameters that were changed in the Alzheimer's patients. So that could suggest that, yes, indeed, we do see defect in DNA repair and mitochondrial function in Alzheimer's patients. So we wanted to dig more into this on the molecular level. So we worked with the hypothesis that DNA repair communicates with mitochondria to ensure healthy aging, and also DNA repair capacity decreases during age, and that will lead to an increased number of genomic stress, which will eventually lead to mitochondrial function, or vice versa. Because it is known that there is a well-characterized communication between the nucleus and the mitochondria. So it is possible that this actually goes both ways. So we started out to look at the consequences for genomic stability when we knocked out mitochondrial function. And what we found was actually a complex genetic instability. We of course found increased oxidative damage, which can cause various types of genetic instability. But we also found that mitochondrial dysfunction affected the DNTP pools, which in addition to genomic instability, point mutations, etc., also can cause replication stress by blocking the replication forks during DNA replication. So essentially, when a replication stress occurs, two things can happen. If it's not repaired, then the replication fork will collapse and the cells will die because these replication stress generates double-stranded breaks. Or they can use a DNA repair bypass system called translesion synthesis, which is essentially non-replicative polymerases that switch the replicative polymerases and then they bypass the damage. That can be error-free or that can be error-prone. So many of them are error-prone, so that generates actually mutations, but the cell will survive. So we hypothesize that this genomic stress that is not bypassed by translesion synthesis, that will actually have an effect on mitochondrial function, and then we will be into some kind of evil circle here if it's not repaired. And this is what the next part of my talk will try to address. So we used a mouse model of translesion synthesis, the Rev1 knockout mouse, in collaboration with Niels de Wind at Leiden University. So these mice show premature aging phenotypes, and here are listed some of them. We don't know whether they have problems with cognitive function yet, but we do know that interestingly they have metabolic disorders. So males get type 2 diabetes when they get old, and females get obese. But they have a shortened health and lifespan. So if the DNA repair or DNA damage hypothesis is true, then we would expect that if we inactivate another major DNA repair pathway, the nucleotide excision repair, for instance, then we would generate more damage to the genome, and that would give a much stronger phenotype. And I'm just going to show actually one experiment from this paper, which shows that when we knock out two major DNA repair pathways, then we actually shorten lifespan dramatically. So essentially these animals, they drop dead. So we went back to the Rev1 model system because it still shows the phenotype, but it's a milder phenotype, which is probably more relevant for normal aging. in collaboration with Linda Bergersen from Oslo here, we actually set out to measure various parameters in these mice. And what we found was that in the brain tissue of the mice, we actually saw decreased respiration, suggesting that there's a lower mitochondrial capacity in these animals. And also, when we looked at the mitochondrial morphology, we could see that in normal cells, the mitochondria, they are nice and even and homogeneous, whereas in the Rev1 knockout animals, they are fragmented and uneven. And that is very characteristic for cells with some problems with either mitochondrial function or cleaning systems. So the data from mice suggested that genomic stress can actually cause premature aging and mitochondrial dysfunction. So the question is, what are the mechanisms underlying this? So we measured several different functions of mitochondria, and in all of the assays we did, from mitochondrial DNA content, membrane potential, ATP, respiration, we always found that there was a difference between the wild type and the Rev1 knockout animals. So some kind of abnormal mitochondrial function. We also saw increased reactive oxygen species in the animals, and at the same time decreased antioxidant enzymes, which could suggest that there's more oxidative stress in these animals. And we also found that PARP1 was induced. PARP1 is an enzyme that is detecting unrepaired DNA damage, and then its substrate is NAD+, it's activated, and then PARP1 is involved in activating the DNA damage response. So if there is a hyperactivation of this enzyme, it suggests that there is unrepaired DNA damage in the genome, and it also suggests that there could be a depletion of NAD+, and I'll come back to this in a minute. Other things that suggested stress in these animals was that if there's an increased energy demand in the cell, AMPK will be activated, and this will eventually inhibit ATP consuming processes and stimulate ATP generating processes and try to save energy. In Rev1, we do not see activation of AMPK, so some kind of defect in this pathway. Another major player in mitochondrial biogenesis is PGC1 alpha. So again, when there's an increased energy demand, PGC1 alpha is increased, which will increase mitochondrial biogenesis. We saw a decrease in Rev1 deficient animal. So to summarize this part, some things suggest that this is the scheme we are actually looking at, where we have some genomic stress, hyperactivation of PARP1. We also saw decreased SIRT1 activity, and etc. So the question is, so now it is known that NAD+ is an important player in this signaling. Is that decreased? And also, these dysfunctional mitochondria, are they accumulated in the tissue because there are problems with mitophagy? So as I said, NAD+, it's known by many, shown by several groups that could actually be one of the link between PARP1 activation and mitochondrial dysfunction. And also interesting is that NAD+ seems to affect all the hallmarks of aging. So that could be a good suggestion actually for a molecule to connect these hallmarks. So we did a metabolomics study in these Rev1 animals in collaboration with Bob Navio at UCDA, University of San Diego, and found essentially two interesting things. One, that Rev1 deficiency changed metabolites that were involved in both nucleotide metabolism and mitochondrial function. But also interesting was to see that NR supplementation actually changed the metabolic profile, both of the wild type, but also from the Rev1 animals, but they never became the same. So clearly, Rev1 deficiency is also changing cellular metabolism independently on NR. But NR supplementation has an effect on metabolism even in wild-type cells. So the cleaning of these dysfunctional mitochondria, as I said, is mediated by a process called mitophagy. It can be either the PINK1 parking dependent pathway of mitophagy or PINK1 parking independent pathway of mitophagy. But mitophagy is ensuring genomic instability. It's involved in preventing accelerated aging and also known to be involved in many age-related diseases like cancer and neurodegeneration. And since we were interested in brain aging, we were looking more into Alzheimer's disease. But before I tell about the studies we have been doing in Alzheimer's, we just went on to ask the question, so is autophagy actually compromised in these repair deficient animals we were looking at? So we measured autophagy indirectly by measuring expression of LCB3, and we also measured mitophagy directly. And what we found was that we both could detect decreased autophagy in the Rev1 deficient animal as well as decreased mitophagy, suggesting that this cleaning system is compromised. Interesting was that when we supplemented or increased in cellular NAD+ levels, we could actually restore or rescue the deficiency in autophagy in the DNA repair deficient animals, suggesting that NAD+ levels actually plays a role in this model system. We tested essentially the same thing using another model, C. elegans, where we could see that after increasing NAD+ levels, we increased both health and lifespan in the Rev1 deficient animals, suggesting that it's an universal regulatory mechanism that there is NAD+ depletion in these DNA repair deficient cells and animals. So we wanted to take this further and look more into whether the same mechanisms are relevant for Alzheimer's disease. So this is our strategy. I just told you a lot about our work in models, cell lines and C. elegans and mice. What I'm going to do now is I'm going to give you a pitch, show you some of our preliminary data from the Danish Alzheimer's, or it's not Danish, they're British actually, from the Alzheimer's patients, where we are studying whether there is a defect in mitophagy in these cells, and also whether we can use NR or NAD+ supplementation to intervene if there are any defects. I should say we don't know this yet, this part. We are right now setting up a clinical trial with the University Hospital of Copenhagen to see whether we can restore some kind of cognitive defects in the Alzheimer's patients. So we don't know yet, unfortunately. But I'm going to just show you a few slides about this part. So we asked essentially what are the molecular alterations that could result in impaired mitophagy in Alzheimer's. And we're also interested to see actually at what stage in the pathology do we see the defect or impaired mitophagy. And then, of course, we are right now trying to see whether interventions with NAD+ can actually have beneficial effects on Alzheimer's patients. But for the molecular part, so in collaboration with Samuel Carter's group at Oxford University, we got some iPSC cells from Alzheimer's patients and age-matched controls. And using his protocols, we can differentiate these iPSC cells into neurons. And when we do that, interestingly, we see actually that both mitochondrial transcription factor and mitochondrial protein, TFAM, and also PDC1 alpha, important for mitochondrial homeostasis, are decreased. We see decreased mitochondrial DNA content and also increased reactive oxygen levels, suggesting that the AD neurons actually show dysfunctional mitochondria. So we also looked at mitophagy in fibroblasts of the Alzheimer's patients. And when we used FCCP to induce mitophagy, we could see that in the controls, we could induce mitophagy in the AD fibroblasts. Whereas in the AD fibroblasts, we could not induce mitophagy, suggesting that mitophagy is actually compromised in the AD cells. And we also see that the lysosome signaling is hyperactivated, suggesting that the AD cells is constantly trying to remove dysfunctional mitochondria or defective organelles. So just to sum up on this part of the talk is that we are working like many other groups are for two strategies that actually it is hopeful to think that we will be able to identify molecules, specific molecules that can actually modulate cellular pathways that are decreased, compromised, dysfunctional in aging and age-related diseases. At the same time, if we can actually identify any genetic defects in these cells, then we will be able to restore the function and then use the iPSC cells to differentiate into different cell types and use them for transplantation. So that is the more translational part of these types of study. But I think that a lot of progress is being doing at the moment. So I'm pretty hopeful that within the not too distant future, we will see a lot of exciting findings from groups around the world. So when we're talking about intervention, a very, very challenging question is, so when is actually the optimal time for intervention? So if you have a lifetime, life course here, so our cells begin to decay, and then one disease pops up, another, and then we will get single mobility, multi-mobility, etc. So when is actually the optimal time to intervene if you want to intervene against these age-related diseases? So one strategy is, of course, to try to intervene before onset of the disease, or at least in the beginning of onset of the disease. So I'm going to, the final part of my talk here will just be a sort of a quick tour through one of the projects that was started in Center for Healthy Aging in 2009. And it's a study, Copenhagen longitudinal study of male cognitive aging. So the goal of this project was to find early predictors of cognitive decline because cognitive decline is a characteristic of dementia, Alzheimer's, and other neurodegenerative disorders. And this is a collaboration in Center for Healthy Aging between myself and Martin Lauritsen, Paul Jenum, Egil Rostrup, and Anne-Marie Lønge-Petersen. And what we were focusing on was to try to investigate whether cognitive performance could be an early predictor, sleep, quality of sleep, oral health, and various molecular functions, bioenergetics, mitochondrial function, which we know is involved in Alzheimer's disease, for instance. So we used a cohort of men born in 1953, and they have been essentially followed from birth. And several times during their lifetime, they took intelligence tests, cognitive tests. First time when they started in the first grade, then when they were drafted for the military, and later on we have actually tested them twice, once in 2010 and recently in 2016. So what we did was that we selected around 200 individuals, either performing extremely well in these cognitive tests or performing extremely poorly in these cognitive tests, because we thought that would be sort of the two extremes, which would be good to look for biomarkers or predictors, especially if there's a lot of variation or there's small differences. So in a number of publications, we showed that, and here showed one of them, that trying to address the question whether sleep quality or lower sleep quality could be associated with cognitive function, it was interesting to see actually in this study that subjects with poor cognitive performance, they actually had lower sleep quality on a lot of different parameters. We also looked at oral health and also found that there was actually an association between poor oral health and low cognitive performance. And we also found that in the blood cells of these individuals, we again could see that at several parameters, there was differences between mitochondrial function of low cognitive performers when you compared them to high cognitive performers. So clearly, it suggested to us that there are some biological pathways that are responding to these differences in cognitive performance, or the other way around, we don't know. We also looked at several other cognitive capabilities in this cohort, but we were not really able to show that there were any evidence really between extreme cognitive decline and these cognitive performances. We found, of course, that there was a lot of correlation between modifiers of the age-related trajectories that had to or could be linked to several lifestyle characteristic, fitness level, cardiovascular disease, diabetes, etc. However, I should say that we are continuing looking into this because there is a lot of variation in these data set, and it's also possible that when we really get more data out from our recent measurements, that we will actually be able to see some more variation here. But at the end of the day, I will just end my talk here, leaving you for a minute with the quote of our Scandinavian fellows here. And the bottom line is that yes, with aging we do see a decline in cells, in tissues, in brain, but there's also hope that we are actually working towards that we can develop interventions and strategies to delay this decay of organs, cells and tissue. And then there's this whole part of this issue that I've not been talking about, that it's also known that our society is actually, so the environment is affecting our health and healthy aging enormously. So be positive is also a part of it. So by that, I would like to thank the collaborators that have been contributing to this work, the people in the lab and various funding agencies. And then I would just say thank you for your attention.
Thank you. You really went through a lot of things, and it is really very important, I think, what you have achieved to do with your Healthy Aging Institute in Denmark. An example for others to follow. I first would like to see if somebody, yeah, Hilde Låge Nielsen.
So, hi, Lene.
Hi.
This is Hilde Nielsen. I have one question, and then I wonder whether you can also elaborate on another one. The first question. A lot of DNA repair defective models, they develop liver disease, but many of them have fatty liver disease. Few have liver degeneration, as in your REV1 defective mice. Do you think that's related to the huge influx of strange chemical compounds in the liver that make sure that you really need these very versatile translation polymerases to cope with these adducts in the liver. So that was the question. And then the second, can you comment on the quite substantial sex difference in the survival plot in the REV1 defective mice? Why do the female mice suffer so much more?
For the first question, yes, I do see that. Do think that it's an increased damage load in the liver cells. I mean, in high proliferating tissue, translesion synthesis is also very important to ensure bypass, to ensure cell proliferation. So I think, yes, that's definitely a part of it. We actually see comprehensive mitochondrial dysfunction in the liver also. I didn't show that really. But the primary phenotype of these mice, especially when we cross them with the NER deficient mice, is the hematopoietic system. It completely collapses. So stem cells are really a target here. For the second question, the sex differences, we don't know yet, but it's quite interesting, and it's not just longevity. We see a difference. It's also for instance the metabolic phenotype, diabetes. So the males get diabetes, but the females do not when they get old, but they get obese. I mean, it's striking. When we got the first mice, we thought that they had sent us some wrong mice from Netherlands, because we saw these enormous female mice and then normal size males. That is something we're investigating now. I think perhaps it could be a hormonal issue. That's at least what we're looking into.
Thank you.
Thanks. Others in the audience here? Yeah, please.
Thank you very much for the talk. My name is Thomas Schmaub. I come from the fan group in the University of Oslo. My question goes regarding one slide where you mentioned that there was no evidence regarding intelligence or cognitive abilities and decline, but there was correlation with illnesses like diabetes, smoking, alcohol, fitness, etc. But that seems to be conflicting to me, because there is research that intelligence also correlates with less heart problems, fitness, and health in general. So that seems a bit conflicting to me, and I want to see if you have any comments on that. Thank you.
Yeah, that's also why we set out to investigate it, and we did not conclude that there is no correlation. We just had to conclude that we could not show any correlation. But as I said, it could be because there's a lot of variation in our data set in the cohort study, and hopefully that will be better when we get the next measurements of these individuals that we did in 2016-17 included in these studies.
Thank you.
Yeah, I also noticed this, and then you had the brain volume also, and that was a protective factor, if I read you correctly. Is that right?
Yeah, that's my understanding, that is known to be a correlated brain volume with cognitive performance.
First of all, thank you for the talk, really interesting. And I am Maria Jose from Bandro fan group. And my question is related to sleep, because you mentioned sleep as an early predictor of in Alzheimer's disease. And I have two questions related to that. One is, do you know what is the percentage of people that are over, let's see, 60 years or a certain age, and they have changes in sleep and they develop Alzheimer, if there is any study related to that? And does sleeping pills helps to delay the development of the Alzheimer disease? I don't know if you know that. Are my questions.
That's very interesting, and thank you for the many good suggestions here for future studies. I don't know whether there are any published or publications on whether poor sleep is associated with Alzheimer's disease. It's very difficult to do controlled studies for those types of questions, and that's also why we did this study. So our cohort at the moment is too young to get sporadic Alzheimer's, actually. So we hope that in 10 years' time, when many of them supposedly will start getting dementia and Alzheimer's, that we can link back really to see whether there's an association between sleep quality and onset of Alzheimer's. What we know now is that there's an association between sleep quality and low cognitive performance. But it's interesting because there are also studies where, for instance, from Mike Nedergaard's group here at University of Copenhagen, showing that sleep is enormously important to clean up the tissue in the brain. So I think it is, from my point of view, it's a logical result that we actually can see already now that there is some association between sleep quality and cognitive performance.
I think at this point I'd like to ask Þóra Káradóttir, if you have a comment or questions.
Well, my only question for Lene would have been, and I don't want to, is whether, well, she kind of mentioned in a talk, with her clinical trials, when to intervene with the NAD+, and whether she has a good idea of how, for the most positive or effective outcome.
That's a good question. We actually don't. We are hopeful. Personally, I think we are looking for small changes in these Alzheimer's patients. But that could be interesting finding to develop strategies actually when to intervene, because I think that's one of the most important questions with these clinical trials. So when do we intervene? When it's too late, essentially? To really generate knowledge about when would be the optimal time to intervene for these age-related diseases. But it's not simple experiments, because it requires longitudinal studies with cohorts, and it's time-consuming, of course. You might be dead before the experiment is finished, but it's also expensive. So, I don't know.
Do you think, sorry, does NAD plus have a way, can you measure the levels? Is it possible to be used as a biomarker?
We can of course not measure it in the brain. We are right now measuring it in blood and CSF. But it's also not black and white here, because it's not like Alzheimer's patients are completely depleted. And with everything else, like when you measure DNA repair capacity, etc., there's also a large variation within the healthy population. So it's small differences, but right now we measure it in CSF and then assume that this is the effect we can see in the brain.
On that note, one thing that I think is relevant, if you give medication to increase the level of NAD+, to what extent do you think there is a danger of having too much?
I think that's relatively small because most of the studies I have seen, NAD+ supplementation doesn't really have an effect unless you have a depletion. Then it's essentially a vitamin, right, or precursor to vitamin B3, so it will be flushed out if you get too much. I think the interesting part is if you have too little, then it seems to have an enormous effect, but otherwise not really.
Very good. We had an additional question here.
Hi, it's me again. I wanted to ask again something, well, not again, but something related to the sleep again. I suppose most of the measurements in the study are being using a polysomnograph in a lab. I wanted to ask if by any chance has been thought to measure the sleep using one of the most advanced trackers that are in the market nowadays to at least be able to have a constant measure of people without them having to go to the lab, because many will argue that having a polysomnograph in a laboratory is not something that would be equivalent to a natural sleep that someone would have in their own house.
Yeah, no, I fully agree. These studies were done at the University Hospital of Copenhagen in the clinical department where they actually see patients with sleep disorders. So it's very controlled environment.
But I mean, in the longitudinal study, is that how often are they being measured?
For sleep, they have been measured twice, in 2010 and 2016.
Oh, okay. Thank you.
But for other parameters, multiple times during their lifetime. But for sleep, only twice.
Would it be possible to do it more often or provide them with a tracker?
But we hope so, but things like that, unfortunately, is associated with funding.
That's true. Thank you. Like everything else.
Yeah.
We do still have a couple of minutes. I see no hands waving. But I noticed that you had very clear observations in fibroblasts that you could take from patients. And of course, these days you can convert fibroblasts into neurons, or astrocytes for that matter. I suppose you have thought about the possibilities in this field for testing whether this is a patient who really has deficient formation of NAD+, and whether it will be someone who can benefit from such treatment.
Yeah, we will. Yeah, definitely. And we are also going to try to set up protocols to differentiate into other cell types like microglia, astrocytes, etc., and compare, because I think it's very, very likely that there is a different response in the different cell types in the brain, because if you just look at, we have also done many global gene expression analysis, and also if you dig into the literature and databases available, you will see that depending on the brain region, cell type in the brain, there's a different expression pattern. So I think it's very, very important to actually look at different cell types.
And oligodendrocytes, of course, would be one of those. And of course, it will be difficult to mimic the different locations in the brain in those cells that you convert in vitro. But maybe there is still in the future possibilities also there. Yeah.
Yeah, we have a pretty large biobank in Denmark containing millions of biopsy samples and also many brain biopsy samples. So we hope also to be able to utilize this biobank to address some of the questions directly on the brain and not always indirectly.
Yeah, we're just on time, so I'm very happy that you have both speakers have been able to come here in this virtual way and contribute something that I think is very, very important. And we look forward to further contacts and to the future where we will be able to see each other in vivo. So thank you very much, and give everybody a hand.